So today's video is going to look at non-Mendelian genetics, so things that are not so simple, just as this one's dominant over this one. So the first thing we're going to look at is what's called gene mapping, and we'll practice with this in class, and we'll work some sample problems together and work with this, but I want you to be familiar with what it is. Okay, so when we're doing gene mapping, we're looking at occasionally genes will be inherited together as a unit. Okay, so far, we've only been talking about things being inherited totally separately, but occasionally things will be inherited together as a unit. They are linked. Okay? And as a general rule, those are going to be the genes that are closer together. So for instance, on this particular chromosome example that you see here on the left, these two are most likely to be linked together, most likely to be inherited together. The closer they are, the more likely they're to be inherited together. Basically, just because if I have a set of genes, then here's another one, and here's another one. Okay? I have a much bigger space between here and here and here and here for crossing over to possibly happen. This is a very small area that would have to be very exact and for crossing over to happen right in the spot in between those two to prevent those two from being inherited separately, to prevent those two from being inherited together. So the genes that are closer together, like I said, they will be inherited together. So we're going to walk through an example of fruit flies and what this looks like with them and calculating what's called crossing over value and mapping the gene. And then, like I said, we'll practice with that in class as well. So what we're looking at here are fruit fly genes, or what is called Drosophila, and it's very commonly used for genetic testing, mainly just because they reproduce very quickly, and they're easy to study, and they're easy to catch, and they're easy to calculate your data from. So what we're looking at here is what's found on the same chromosome. And so we're going to be looking here at wings. Okay, so you can see genes for wings there. We're going to be looking at leg length, body color. Okay, we're going to look at all of these things. Occasionally we look at eye color with this. Okay, but we're going to be looking at which one of these are inherited together. All these things here are on the same chromosome. So you can see we've got a sample there of what's on that particular chromosome. And we're going to run some test crosses to see if they're linked together or not. We're running this test cross here, and remember, your test cross, you're linking a known with an unknown, and so we're doing this to see, like I said, we're doing this to see which genes may possibly be linked together. So in this P generation, in this parent generation here, we're going to be crossing what's called a wild type, which a wild type has alleles for a gray body, and you can see those are represented as B+. And then alleles for what are called normal wings. So the plus sign there indicates that it's a wild type fly. And so we're going to cross that with a homozygous recessive, and this is considered a mutant. Okay, so this homozygous recessive mutant. So it has a black body here and what are called vestigial wings. And you see these wings are significantly smaller than those wings. If you remember from evolution, your vestigial structures were usually shrunken in size. Okay, so we're doing this test cross here, and we're going to examine, and we're going to see that all of the F1 generation, they're all going to be exactly the same. So you can see your F1 generation down here. Okay, so my F1 generation is all going to be B plus B, as well as VG plus, and then VG. And getting one from each parent. So now using that F1 dihybrid generation, so this one here, we're going to do another test cross, again with a double mutant. Okay, so first we crossed these two guys and ended up with that one. All of the generation will be like that. Now we're going to add this double mutant in again to cross it with the dihybrid, to cross it with the F1 generation. When we cross that F1 generation with the mutant, we end up with, this down here. These are our possibilities here. And so we've taken all of our possible combinations of eggs from the female, all the possible sperm combinations from the double mutant, okay, and basically done a minimized Punnett square here. So we have these four possible outcomes. Okay, and so typically we would, if these genes were on different chromosomes, we should see even numbers of each one of these. We should have a nice 1 to 1 to 1 ratio if these, again, were on different chromosomes. Okay? But what will happen is that's not what we will actually see. Okay? We're going to see uneven numbers, okay, which is going to show us that there's crossing over occurring and that the genes are linked together. Here's the actual results, and you can see that these alleles, we've got uneven numbers down here, okay, which again shows linkage between some of these genes. So these genes are not on different chromosomes, and that some of them are inherited together. It is way more common, okay, to see these two than to see the other two combinations of genes. This also shows that parental alleles are often linked together as well, inherited together. And you can see here in your description, it's telling you that. If you follow this all the way back to the beginning, we had our original wild-type fly, okay, which had its B plus, VG plus that it passed on. So it passed on this to the dihybrid, okay, whereas the mutant passed on those alleles. So if those alleles are continually inherited together, so as this female here, okay, and her alleles are being separated into eggs, these genes are linked together, as well as the parental alleles are inherited together. So all the wild type alleles, it's more common for them to be inherited together, as well as more common for the mutant alleles to be inherited together. And so that makes those uneven numbers makes those first two combinations a lot more prevalent. Okay? And so what this will do is we can actually we can use what's called a crossover value, and we can assign a frequency number to how often we think this will occur. Okay? A little vocabulary real quick though before we do that. Okay, just to be sure that we know and are comfortable with what it means to be a recombinant. Okay, so a recombinant is really just going to be new combinations of alleles. And so in this case, we do have two recombinants, okay, which are brand new combinations of alleles, which is your B plus VG and then your B VG plus. So those are recombinants. You can see that here. Those new combinations of alleles, which would come from the crossing over and the various things that help ensure genetic variation that occurred during meiosis. Let's use this example here, and we'll use it to calculate what's called our crossover value. And so our crossover value is going to be our number of recombinants. Remember, those are our new combinations of genes. So the number of recombinants over the total number of offspring. So if I look at this particular example here, okay, I've got my number of recombinants, which is this right here. Okay, those are recombinants. These are the ones where I got my new allele combinations, right, that I didn't have before. Okay, I had these before. This was my F1 generation dihybrid. This was the mutant. So these are brand new allele combinations, so my recombinants. When I add those together, I get 391. Okay, and then I divide those by my total offspring. So adding all of this together, okay, I've got 2,300. Okay, and when I divide those, I get 0.17. And if I multiply those by 100%, I get 17%. So that's going to be my frequency. That's what's called my crossover rate. Okay, and so basically this is going to tell me, it's going to give me an idea of how close together these genes are. The higher the frequency, the farther apart they are. Okay, and so this one at 17% tells you that they're relatively close together and they're likely to be inherited together. Crossover values or crossover rates can be converted to what are called map units, and that's the gene mapping that we'll practice in class. Okay, and so a map unit, one map unit equals one crossover percentage. So for instance, in this case, okay, if I've got 17% as my crossover rate, then these two genes are 17 map units apart. Okay, and so again, this just gives us a quantitative value and helps give us an idea of how far apart that we believe these genes are. And again, the larger this number are, the farther apart they are, which means the less likely they are to be inherited together. Okay, so let's do a little freshman year review as well, real quick. So some of this you guys should be familiar with. So let's start first with what's called complete dominance. Complete dominance is kind of your traditional genetics problem. It's the one you probably did the most of your freshman year. Complete dominance is what we've already talked about. This is when your phenotype of your heterozygous looks the same as your homozygous dominant. You know, if I said that somebody had brown hair was dominant to blonde hair, and so these people have brown hair and these people have brown hair. Okay, and so the phenotypes of both of them look the same. So let's look very quickly again at incomplete versus co-dominance. Okay, so what we have here is incomplete dominance happening. Okay, and so with incomplete dominance, you're going to see the phenotype of the hybrids is going to be somewhere in between the two parents. Okay, so here's my parent generation up here. Okay, and so the alleles here are CR, CR, and CW, CW. So this one passes on a CR, this one passes on a CW. Now my F1 generation is going to be these hybrids that are CR, CW, okay, and they're going to look pink, which is going to be what looks like a combination somewhere in between the phenotypes of the two parents. And so half of the gametes that'll be passed on will be the CR, okay, half of them will be the CW. So now in my F2 generation, I get the possibility of all kinds of offspring here. Okay, I've got the possibility of red offspring that have both CR alleles, pink offspring that have CR and CW alleles, as well as white offspring, which have to be CW, CW. Okay, so in incomplete dominance, basically your heterozygote gets your hybrids get what looks like a combination phenotype between the two parents. And the other thing that we have is codominance. And codominance is usually a result of multiple alleles. So there are two dominant alleles, and they both are going to affect the phenotype. Okay? So there's at least two alleles that both affect the phenotype, and they affect it in a manner that is very distinguishable. It's noticeable. And our most common example for codominance is going to be blood typing. That's the one you should have seen your freshman year. So that's the example we're going to stick with. So let's kind of review that for a minute here. Remember, with our blood groups, we had 3 alleles for blood groups. Remember, we had an IA, an IB, and a little I. And these were all, IA, IB are both dominant. Okay? And remember, they produce the carbohydrates. Remember, these produce the antigens. Remember, these surface markers are antigens, and they allow for the body to be able to recognize which kind of cell it is. And so the allele for IA produces the A surface marker. The allele for IB produces the B surface marker. And the recessive allele, the little I there, doesn't produce any surface markers. Right? And so when we look at the genotype versus phenotype with these, so going across the top here, I'm going to see my genotypes. Okay, this is what the red blood cell will look like. So its appearance is going to be in this row here. And then down here on the bottom, I'm going to put the phenotype. And phenotype for blood groups is your actual blood type. Okay, and so in this case, if I look at this cell here, I have A surface markers, or A carbohydrates, which means I'm talking about blood type A here. And the genotypes that I can get for blood type A are big IA, or big IA, little I. So I can be heterozygous, like a typical dominant situation, one dominant, one recessive. okay, or I could be homozygous dominant. And the second little cell there, okay, it's got B surface markers, so that means it must have the B allele. Okay, so the genotypes possible here are IB, IB, as well as IB little I, and so the phenotype would be type B blood. And then if we look at the AB blood group there, this cell right here has both A and B surface markers. So it's affecting that. Doesn't look like a blending. I didn't get a surface marker that is like half triangle, half circle. Okay, I didn't get that. I got distinct circles, distinct triangles. So I got phenotypes that are showing from both alleles in a separate, distinguishable manner here. So that means I have to have an IA allele to be able to produce those those triangle-shaped surface markers there, and I have to have an IB. Well, we only get one allele from each parent, so that's it, I'm done. There's no other genotype. Somebody that has type AB blood cannot have any recessive alleles because they don't, if they had any recessive alleles, then they wouldn't be able to have both of the dominant ones. And then down at the end, we have the final one, type O blood. Okay, type O blood, there's no surface markers on those red blood cells, which means the only alleles they can have are little i, because that's the only way they do not produce any surface markers. And remember, we can put these into a Punnett square. Let's run through an example problem real quick where we would see that. So let's say I have a mom who is type O and a potential father, okay, and then a baby who is type O, and then a potential father is type B. Okay, and so we're going to see if that would work and if that was possible for Ira to be the father, for this this guy here to be the father. Well, if we think about it, if mom is type O blood, then mom has to be little i. If the potential dad is type B blood, we know he has to have at least one IB. Okay, he's got to have at least that. So if I am plugging this into a Punnett square to see if they can produce a child that's going to have also little i. Okay, well, mom is little I. Dad is at least IB. So these first options here don't give me any option. Okay, those don't work, those don't work. But remember, if we go back to our genotypes, the possible genotypes for type B blood, they can be homozygous, IB, or they could be heterozygous. So if he is heterozygous, then he has the definite possibility of producing a child with type O blood. So it is possible that he could be the father. In addition to the various types of dominances, we also have what's called polygenic inheritance. Okay, and so polygenic inheritance is basically it's an additive effect. There's two or more genes that are impacting a particular phenotype. Okay, it has what are called quantitative characters, and these are things that are continuous along a broad spectrum. Okay, they vary in the population along a particular continuum. If we look at the example here that we've got is human skin color. That's an example of polygenic inheritance. Okay, and so you can see there's a wide variety of skin tones. Okay, and so there's two or more genes. There's like 8 genes here that impact skin coloring. And so this is an additive effect. So the more of these alleles that you have, the darker the skin coloring is. And so the less of these alleles that you have, the lighter the skin coloring is. And so this would be an example of polygenic inheritance. We've got this additive, this cumulative effect happening because there's more than one gene that contributes to the particular phenotype. The thing we need to take into consideration is the environment. It can have an impact on the genotype's expression, the phenotype that the genotype will show us. And that's what's called our norm of reaction. Okay, so our norm of reaction is a phenotypic range that a particular genotype will have based on environmental influence. So the environment can only influence so much, but it can have an impact on some phenotypes. The example we see here are these hydrangea plants. Okay, and hydrangeas can either be blue or pink, and that's going to depend upon the soil. Okay, blue hydrangeas actually are in soil that have a lower pH, so it's more acidic, and so they start getting this bluing to them. Pink ones are in a soil that has a little bit of a higher pH, okay, so it's a little bit more basic, a little closer to neutral, okay, and so they will not do as much of the bluing, and so they will appear pinkish colored. Okay, so just this, we're just looking at some things other than just straight Mendelian, this is dominant over this genetics, looking into some other factors that can contribute to these various phenotypes, and we'll work with these as well in class.